All right. Are there any questions? Any questions up to whatever we covered last time? Right. So this was, I believe, the slide that on which we stopped. Right. So one application of DNA hybridization is in the prenatal diagnosis of genetic diseases, for example, sickle cell anemia, right? So before a child is born, you want to know whether that child is going to have sickle cell anemia or some other disease, right? And that can be done using today's DNA technology, right? Now, examining a single gene in the human genome requires searching through a total genome of over 3 billion nucleotides. That's a big task, all right? But this can be simplified because we know exactly if a child or a person is going to have sickle cell anemia, we know exactly, right, what DNA mutation is going to cause that disease, right? So we can go looking for it in a very, very precise fashion. And I'll give you some details about that in a few minutes. Yeah, here there's a picture, that is basically showing the denaturation and the renaturation of DNA molecules. So DNA always occurs in nature as a double-stranded molecule, right? And you have these DNA bases forming the rungs of this ladder, right? A is always linked to through how many bonds? 2 hydrogen bonds, right? And G is linked with C, 3 hydrogen bonds, okay? And this AT and GC linkage makes sure that the width is the same all throughout, right? So you have this kind of a staircase, right, that spirals around itself. But if you subject double-stranded DNA to high temperature or a high pH means what? Very? Yeah, yeah, that's right, yeah. pH less than 7 is acidic, pH more than 7 is basic. Then it will denature the DNA. The double-stranded DNA becomes like single-stranded, right? But then when you remove this, right, you either cool it or you lower the pH, you make it more acidic, right? Then again it forms a double helix, right? And DNA is a very stable molecule. That's why in criminal investigations and all that, if you leave behind a sample of hair or something, they can extract the DNA from there and they can use that to nail down the suspect, right? So no matter to what lengths criminals might go to, remove all traces, with the DNA you can kind of track it down. And in fact, probably later on in today's lecture we will talk about that. How do you track down suspects? How do you nail down suspects in a criminal investigation? So, as I mentioned on the earlier slide, right, looking for like a single gene among the three or single nucleotide among the 3 billion nucleotides is a big task, okay? However, the thing that we have going for us is that if you go looking with the complement of the DNA sequence that you're interested in, right, the, the two bind with great specificity, right? So that's something that you have going for you, right? So the tremendous specificity of DNA hybridization makes it possible to do so in a fairly tractable fashion, to look for a specific DNA sequence even among the 3 billion nucleotides. For example, for sickle cell anemia, the exact nucleotide change in the mutant gene is known, right? We know, right? Because you just change one nucleotide, the amino acid changes, right? And you get sickle cell anemia. So if you're going to do prenatal diagnosis of sickle cell anemia, DNA is extracted from fetal cells, right? And you construct two DNA probes, right? So two DNA probes are used to test fetal DNA, one corresponding to the normal gene sequence in the region of the mutation, and the other corresponding to the mutant gene sequence, right? Now what is a DNA probe? A DNA probe is a short, single-stranded DNA. An oligonucleotide. Well, what is an oligonucleotide? An oligonucleotide is a synthetic nucleotide, right? Because like today we have the capability to engineer DNA molecules in the lab, right? So we can go and synthesize that. So that would be an oligonucleotide, a short stretch of DNA, which is used in hybridization reactions to detect nucleic acid molecules containing a complementary sequence. So if you're looking for a particular DNA sequence, go ahead and artificially synthesize the complement of that sequence, right? And then use that as a probe, because that is going to hybridize to the sequence that you're looking for with great specificity. Right? That's the key idea. So how is this procedure implemented? So DNA samples from the fetus are first treated with restriction nucleases, right? So what is the restriction nuclease going to do? When you treat the DNA with restriction nucleases, it is going to? It's going to what? It's going to break it? break it down, right, into much smaller pieces in a very, very predictable fashion, right? So DNA, because you're not going to deal with those 3 billion nucleotides, you want smaller pieces, right? So you're going to take DNA samples from the fetus, treat them with restriction nucleases, and all the resulting DNA fragments are then electrophoresed through a gel. What is electrophoresis going to do? It is going to separate out the DNA fragments on the basis of their size, because depending on the length of the segment, all right, of the fragment, it's going to move a different amount, all right, in that gel. So the gel is then treated with a DNA probe that detects only the restriction fragment that carries the beta globin gene, right? Because you know the exact mutation, right? So you went ahead and constructed the complement, right? Right? So you created that probe. So using the two DNA probes, so one DNA probe will correspond to the normal gene, right, in that region. Another DNA probe will correspond to the sickle cell gene in that region, right? So using those two DNA probes, it is possible to distinguish whether the fetus contains one, two, or no defective beta globin genes. Because remember, I said before that in order to get full-blown sickle cell anemia, you need two defective beta globin genes, right? And you have two copies of that gene, right? So there are there are a number of possibilities. Either you have no defective gene, right? You have one copy of defective gene, in which case you become a carrier for the disease, right? Or you have two copies of the defective gene, in which case you have full-blown sickle cell anemia. So there are three possibilities, right? So using these two probes, you can actually examine, okay, does this cell have two defective beta globin genes, okay, or only one defective beta globin gene, right? Or no defective beta globin genes, right? You can figure that out. And the lab procedure that is used to visualize this hybridization, it's known as southern blotting, right? So in molecular biology, you will see these terms: western blot, northern blot, southern blot, and so on, okay? Procedure is similar, right? It's just that in different contexts, they have been given different names. I don't know what the reason is for calling this the southern blot, okay? But what are the steps involved? First, you cleave double-stranded DNA using restriction nucleases. That's what we talked about at the top of this slide, okay? Now you're going to electrophorese the fragments to separate the fragments by length, right? We already talked about that before, right? And this is possible because DNA is negatively charged, right? So in an electric field, it is going to migrate, right? Then you want to capture this position information, right? So how do you capture that position information from the gel? Well, you put a sheet of nitrocellulose paper, right? It's like blotting paper, right? Is laid over the gel, and the separated DNA fragments are transferred to the sheet by blotting, right? So now you have all the position information, right? And these fragments have been separated out, right? So now you're going to peel that paper off, right? Well, before that, you know, as the DNA fragments are transferred onto the sheet, right, the DNA gets denatured, right? That means the double-stranded DNA becomes single-stranded, right? And the single-stranded fragments, they stick firmly to the surface of the nitrocellulose sheet, right? So now you peel off that sheet, right, from that slab of agarose gel, right? And you will take this sheet, right, put it inside a sealed plastic bag, like maybe like a Ziploc bag, right, that we use every day, right? So the sheet containing the bound single-stranded DNA fragment is placed in a sealed plastic bag together with buffer containing a radioactively labeled DNA probe specific for the required DNA sequence, right? Because you know the exact region of the mutation. So you've created those probes. You can label them radioactively by putting, you know, radioactive phosphorus, right, in that probe. So once you mix them up, right, now if there is a perfect match, right, the probe is going to hybridize to some location on that sheet, right? So this gives the probe a chance to hybridize with its complement if the latter is present on the sheet. Then you remove the sheet from the bag. You wash it thoroughly. So wherever there is no hybridization, it's single-stranded, everything is washed off, right? Only the hybridized stuff remains, right? So after autoradiography, the DNA that has hybridized to the label probe will show up as bands on the autoradiograph, right? And if you have, you know, two defective beta globin genes, all right, you will get both the bands will be in the same location, right? If you have one defective, one that is normal, you'll have them separated, okay? If both are normal, again, they'll be in the same location, but somewhere else on the sheet, right? And you have prior information about that. So using this technique, you can figure out whether the child that is going to be born is going to have full-blown sickle cell anemia, right? Or going to be a carrier, or is going to be normal. All right, any questions? And again, all these processes that I'm talking about, they are easier for us. They're easier said than done, because putting it on the blotting paper and all that, they have protocols, okay, that the biologists in the lab, and that's why their work is so very labor intensive, right? It's not like us, you just sit with some paper and you write some equations and do something, right? It's labor intensive. They have to follow the proper protocols to make sure that they don't get, because if you have garbage in, you'll get garbage out, so you have to follow the proper protocols. We cannot get into that, because we are not the biologists. Let them do that work, all right? But if we have some understanding, all right, of the big picture, we are in a much better situation to interact with them. Okay. So next I move on to the topic of DNA cloning. DNA cloning has got two possible meanings in biology. All right, the first meaning is the act of making many identical copies of a DNA molecule, right? And the second one is the isolation of a particular stretch of DNA from the rest of the cell's DNA. And this isolation is facilitated by making many identical copies of the DNA of interest. Thereby, you're amplifying it relative to the rest of the DNA, right? So in both cases, you are actually making many copies of that DNA, right? So just as restriction nucleases can be used to break DNA into smaller fragments, right? An enzyme called DNA ligase, which we have already encountered earlier in the context of DNA replication. Remember? So why did we need DNA ligase in the context of DNA replication? Anybody remember that? Exactly, yeah. Thank you. So this DNA ligase can be used to join DNA fragments together to produce what is called a recombinant DNA molecule. And you might have heard this terminology, okay, recombinant DNA technology. This is what it means. Cut up pieces of DNA from somewhere else and then combine it together, right? Stitch it back together, you get a new DNA molecule. Now since DNA has got exactly the same chemical structure, and we know that, having taken this course, we know that right now, okay? You're going to have the base, you're going to have the sugar, 5-carbon sugar, then you're going to have the phosphate, all right? So since DNA has got exactly the same chemical structure in all organisms, the use of this enzyme, that is DNA ligase, allows DNA from any source to be joined together, right? You can take like DNA from a dog and human DNA and join that together, right? So restriction nucleases and DNA ligase play an important role in cloning DNA. And DNA cloning can be carried out using what is called a bacterial plasmid. A plasmid is a small circular DNA molecule. We have seen a plasmid in the context of what? Yeah. Bacterial plasmid, yeah, the fertility plasmid, we talked about that. Maybe last lecture or the lecture before that. So how do you carry out DNA cloning using a plasmid, right? So the purified plasmid DNA, right, it's a small circular DNA molecule, is exposed again to a restriction nuclease. So you're going to cut it open, right? That cleaves it in just one place. And the DNA fragment that is to be cloned is covalently inserted into it using DNA ligase, right? Because now we know how to cut DNA using restriction nucleases. We know how to stitch DNA back together using ligase, right? So the DNA molecule that you want to clone, right, you're going to insert it into the plasmid by first using a restriction nuclease and then using ligase, right? Then this recombinant DNA molecule that you have, right, is then introduced into a bacterium, usually E. coli, by transformation, right? We know how bacteria can take up DNA from other organisms, right? So by transformation you put it in, right? And there are protocols that have to be followed, all right? And then once you have it inside the bacterium, you allow the bacteria to grow in the presence of nutrients, right? And every 20 minutes, the bacterium is going to divide, all right? Every time the bacterium divides, even the plasmid information in the plasmid also is going to get replicated. So after just a day, more than 1 billion copies of the plasmid would have been produced, right? So the bacteria then lyse, so then you have to kill the bacteria. So lysing means to split, right, the bacteria. And the much smaller plasmid DNA is purified away from the rest of the cell contents, including the large bacterial chromosome, right? And the DNA fragment can now be recovered by cutting it cleanly out of the plasmid DNA, again using the appropriate restriction nuclease, and separate it from the plasmid DNA by gel electrophoresis, right? By using the size and all that stuff, right? Okay. Again, I mean, if you or I were asked to actually do these experiments, there's a lot of new additional information that we will need, right? What temperature, okay, under what conditions you have to do the experiment, right? How much time should you allow and so on, right? We are not going to worry about that. We just want the big picture. So any questions on this DNA cloning using bacteria? Now human genes can be isolated by DNA cloning, right? So for example, dealing with the unfragmented 3 billion nucleotides of the complete human genome is a formidable task, right? It's a lot of information, okay? So so you don't have to deal with that. You can avoid this situation by breaking up the total genomic DNA into smaller, more manageable pieces to make it easier to work with. To do that, the total DNA that is extracted from a tissue sample or a culture of human cells is cut up into a set of DNA fragments, again by restriction nuclease treatment, because that will cut it up in a very, very specific, predictable, all right, and repeatable manner, right? And then each fragment is cloned using bacterial plasmids as described earlier, right? I told you how you can clone a fragment of DNA. Now the collection of cloned DNA fragments thus obtained is called a DNA library. Right? You might have heard this term too, right? DNA library, cDNA library. So this is the DNA library. So in this case, this library is called a genomic library. Right? It's like a library with many books. You have all these clones of different genes, right? And these are commercially available. You can buy them in the market. Right? So in this case, the name is genomic library, as the DNA fragments are derived directly from the chromosomal DNA, right? However, you have a problem. For the library, you need a catalog, right? I mean, if there are lots of books over there, you're looking for a particular book. How do you find that out, right? So here also, if you're looking for, you're interested in the clone of a particular gene, how do you find that out? Any ideas? So you have a library, so you have a genomic library where you have clones of many different genes, okay? How would you find out which library, right, corresponds to the gene that you are interested in? Exactly. Use the complement, Go looking for the complement. And the problem is that you may not, be able to zero in on one clone, because when you split up that gene, the gene might have been split up into different pieces, okay? So they are in different clones. But at least the few clones associated with that gene, you will identify by using the complement. Yeah, that's the idea. So that's what we'll talk about on the next slide. So another thing, while you're producing a genomic library, you have to make sure that each colony of E. coli is producing clones of only one DNA fragment, right? So you want to make sure that each E. coli is taking up only one plasmid, right? Each plasmid is taking up only one DNA fragment, because you want separate clones for each of these DNA fragments, right? So this can be done by carrying out the entire procedure under the following favorable conditions. DNA fragments are inserted into plasmid vectors under conditions that favor the insertion of one DNA fragment for each plasmid molecule, right? And then these recombinant plasmids are also mixed with a culture of E. coli at a concentration that ensures that no more than one plasmid molecule is taken up by each bacterium. This is what I meant, that you have to follow protocols, right? If I just go ahead and just try an experiment, probably it'll be all messed up, right? There's practice needed, right? So a natural question that now comes up is, how can one find a particular gene in this huge genomic library? And as Sitanshu pointed out, right, one way is to go looking. So if you know the sequence of the gene, you can create the complement and then go looking for that, right? So if the sequence of the complementary DNA is known, one could make a probe and use it to identify the particular gene by exploiting the properties of nucleic acid hybridization. If on the other hand, you don't know the sequence of the gene, right? You can use protein sequencing. Like, remember we talked about protein sequencing in an earlier chapter? Like, you try to find out what are the amino acids. You don't have to sequence the entire protein. If you get like a few of the amino acids, if you get the sequence, then you can use that to make a probe, right? Even if some portion of the gene hybridizes, you'll be able to pick it up, right? So by using the genetic code in reverse, again, genetic code is telling you how you go from RNA to amino acids, all right? So by using the genetic code in reverse, the DNA sequences that code for these amino acid sequences can be deduced, and a suitable DNA probe can be synthetically prepared. So you can prepare an oligonucleotide, right? Synthetic DNA probe. So using this probe, those rare bacterial clones containing human DNA complementary to the probe can be identified by DNA hybridization. Okay. Now it is possible, as I said before, that for a given gene, several clones may be identified, as no single clone might contain the entire gene. Right? Because you've cut it up using restriction nucleases, right? So the gene might have been spread across different fragments, right? And this is especially true for long DNA sequences which have lots of introns in them. Again, what are introns? They are the non-coding sequences, or DNA sequences, and they are found in what kind of cells? Huh? Eukaryotes, yeah, good. I'm trying to prepare you a little bit for the exam also. Okay, Now, so we already talked about genomic library, right, which is made up of the DNA. So you take up the DNA of the organism, break it up into pieces, and then for each piece or each fragment of DNA, you create a library, right? That's the genomic library. Now, for many applications of DNA technology, it is advantageous to obtain a clone which contains only the coding sequence of a gene. That is a clone which lacks the intron DNA, right? So it is a relatively simple matter to isolate a gene free of all its introns. For this purpose, a different type of library, which is called a cDNA or complementary DNA library, is used, right? And these cDNA clones, they are sold commercially, right? If there's a particular gene you're interested, they'll ship it to you. You just pay them the money, they'll ship it to you. So the creation of a cDNA library involves the following steps. First, the starting point will be the messenger RNA. Because in the case of eukaryotes, all right, you know that the messenger RNA has all the introns taken out, right? So only the coding region is there. So the starting point is the messenger RNA that is expressed in a particular tissue or cell culture. Expressed means that the DNA is basically being transcribed into messenger RNA, right? So first, starting from the mRNA that is expressed in a particular tissue or cell culture, you construct the complementary DNA, or which is called cDNA, using reverse transcriptase, right? Because it's doing reverse of transcription. Then you degrade the mRNA copy using an alkali or a base and use the single-stranded cDNA that is left over to produce a double-stranded cDNA copy of the original mRNA, right? Right? I mean, once you have like one of the strands, you can make a complementary copy, right? Using either DNA polymerase, or some other procedure, right? The cDNA molecules can then be cloned. So starting from the messenger RNA, now you have the genomic DNA but without the introns, right? For that particular tissue. Now you can cut it up using restriction nucleases, right? Get the fragments, put them into plasmids, right? Right? Then put the plasmids into E. coli, then let the thing grow, right? So follow the same procedure as before, but now the clones that you are going to get, those are actually cDNA clones, and this is going to be a cDNA library, right? So based on the discussion that we have had so far, we can highlight the following differences between DNA clones and cDNA clones. The DNA clones are the genomic clones. They represent a random sample of all of the DNA sequences found in an organism's genome, while cDNA clones contain fragments of only those genes that have been transcribed into messenger RNA in the tissue from which the RNA came, right? So since cells of different tissues produce distinct sets of RNA molecules, right? A different cDNA library will be obtained for each type of tissue. So you'll have a different cDNA library for, let's say, the liver versus the kidney versus some other organ, right? The other difference to note is that the genomic clones from eukaryotes, they contain large amounts of repetitive DNA sequences, right? Introns, gene regulatory regions, and spacer DNA. Because as I have been saying before, only about 2% of the entire information in the genome really codes for proteins, right? So most of it is other stuff, it has other functions. And on the other hand, if you're looking at cDNA clones, they contain only the coding sequences. So thus, cDNA clones are particularly well suited for deducing the amino acid sequence of a protein from the DNA, right? Because all the introns have been taken out, right? So now all you have to do is just go and use the genetic code, okay? Read the nucleotides, the RNA nucleotides 3 at a time, and you know what the amino acid sequence of the protein is. They are also very useful for producing the protein in bulk by expressing the clone gene in a bacterial or yeast cell. Remember, the bacterial cells, the bacteria do not have the splicing mechanisms in place, right? So if you are trying to produce some protein, if you just go and take the relevant human gene, right, and give it to the bacteria, it's not going to work because bacteria do not have the mechanism for cutting introns out, right? On the other hand, if you take stuff from a cDNA library, right, with all the introns removed, you can put it in a bacterium and you can force the bacterium to make many, many copies of that protein of interest, right? Now we should note that I said, hybridization is very specific. If you go go looking for a gene, if you take its complement as a probe, there's going to be perfect hybridization matching, all right? But even when there is not a perfect match, hybridization does take place, all right? So hybridization allows even distantly related genes to be identified. Hybridization can be carried out under conditions that allow even an imperfect match. It doesn't have to be a 100% match, right? Between a DNA probe and its corresponding DNA to form a stable double helix. So this can be used to identify closely related genes using a single DNA probe, or even use a DNA probe for one species to identify the corresponding gene for another species. For example, if I know let's say in mice, okay, I figured out or mice maybe is not a good example. Let's say in dogs, I figured out the sequence of the, let's say, globin gene, beta globin gene, right? So I know what the sequence is, and I don't know where in the genome in the human genome the beta globin gene is. Okay, I can use the complement of that, canine beta globin gene, right, and see where it hybridizes, on the human genome, right, and that would be the location of the beta globin gene in the human genome, right. So things like that have been done. All right, any questions? So we have talked about cloning DNA, which basically means DNA amplification, making many, many copies of it. There is another widely used method for amplifying DNA, and that is called the polymerase chain reaction, right, or PCR, right. So when you hear a biologist talking about PCR, you know what it is, polymerase chain reaction, right. So polymerase chain reaction, or PCR, is a synthetic procedure that can be used to selectively replicate a given nucleotide sequence quite rapidly and in large amounts from any DNA that contains it, right? And each cycle of the PCR reaction consists of 3 steps as shown in the figure. So let's look at the figure. This is much easier to explain using the figure. So you have this double-stranded DNA here. You want to make many copies of this DNA, right? So what you're going to do is initially you're going to heat this DNA to separate the 2 strands out, right? Now then each one of these strands can now serve as a template. You're going to put in a primer to bracket the region that you're interested in, right? So there's a primer here, there's a primer here, because DNA replication has to be started with a primer. Then you add DNA polymerase, because that's the enzyme that is going to synthesize DNA by looking at the template, and then you add this deoxy ATP, deoxy GTP, deoxy CTP, deoxy TTP. So DNA synthesis is going to take place, right? So the complementary strand has been synthesized here, the complementary strand has been synthesized here, right? This is the first cycle, right? So now, I mean, forget about the little bit of DNA that's left out here, right? Let's not worry about that. But leaving that aside, you have basically double the amount of DNA in one cycle, right? In one round of this procedure. Again, in the next round, what do you do? If you want to make more copies of the DNA, you want to amplify it more, again apply heat to this. Separate the 2 strands out, put primers, again amplify. So if you do this n times, how many copies are you going to have? 2 to the power of n, yeah. So you can see if you do it 40 times, even from a small amount of DNA, you would have created a lot of DNA. And you need more DNA to run procedures, because these different procedures. For example, if there's a criminal, somebody has committed a murder or something, they're not going to shave off their hair and leave a lot of DNA for you to go and catch them, They're not going to do that, right? And you might just get 1 strand of hair, right? So, but in order to run a lot of these procedures like gel electrophoresis and all that, you need to amplify the DNA, right? So you'll subject it to this procedure. Now the problem is, if that original DNA is contaminated with some other stuff, right, even that is going to get amplified. So that's where the lawyers will come in, okay, and they'll argue that, okay, this is not this person's DNA, that lab is, contaminated stuff and all that, So as I showed in the figure, in the first step, heat is applied to separate out the 2 DNA strands of a double-stranded DNA molecule. In the second step, primers are hybridized on the 2 strands to mark the beginning of the regions of DNA to be amplified. I showed that to you on the diagram. In the third step, DNA polymerase and deoxyribonucleoside triphosphates. Why do you have to add triphosphates? Right. Yeah, because the reaction that you want to take place, right, that's an energetically unfavorable reaction, right? So triphosphates are added so that DNA complementary to each of the 2 strands and originating at the 2 primers can be synthesized. So in one cycle of PCR, the quantity of DNA is amplified by a factor of 2, right? And you do this n times, you'll get 2 to the power of n. So this is the first cycle, second cycle, third cycle, and so on, right? So if beyond the first cycle, if you need additional amplification, just repeat the procedure over and over again. In n cycles, you would have amplified by a factor of 2 to the power of n. Now in each cycle of the PCR reaction, you are heating the DNA to separate the 2 strands, right? So if you use regular DNA polymerase, all right, that the polymerase will get destroyed. So in every cycle, you have to keep on adding new DNA polymerase, all right? So so instead of doing that, the DNA polymerase that is used for PCR is a special kind of heat-resistant polymerase, which is usually isolated from heat-loving bacteria, like thermophilic bacteria that live like in these hot springs and in the vents of volcanoes and all that, you know. So they have DNA polymerase that is resistant to heat. So we are next going to describe some common applications of PCR, right? What are you going to do once you have amplified the DNA? Okay, what are some of the applications? Now, before I finish this topic about PCR amplification, I should just mention that there is a professor here in chemical engineering, right? His name is Victor Ugas, all right? And he works on this kind of stuff, on this PCR, right? And he's trying to use, see, because every time you have to heat the DNA to separate the two strands out, right? So he's trying to use like, principles of convection, okay? Because if you have convection, you're moving from a hot region to a cooler region, right? So he has some results on that. You know, I remember about 10, 12 years ago, a student of his was taking this class, you know, and he told me about it. And that professor is here in chemical engineering, so he works on that. So it's like you're trying to basically, use principles of heat transfer, right, to make this procedure more efficient. I don't know the details, but it's basically by using convection, right? Because it's a heating and cooling, right? I mean, every time you're trying to separate the two strands and you have to bring them together, you have to cool, right? So he's used some convection, he's introduced some convection to make the procedure more efficient. So now let's look at some applications of PCR, right? So PCR can be used to clone directly a particular DNA fragment. And the main advantage of PCR is that you don't have to do any cell culturing, right? Because earlier on we were saying that you'll take that DNA fragment, then you'll put it into a plasmid, then put it into a bacterium, grow the bacterium. So you have to do cell culturing. Here you don't have to do any of that, right? So no cell culturing is involved. Now, one of the applications of PCR is that it can be used to detect viral infection at very early stages, okay? Because at very early stages, the amount of viral DNA or RNA is going to be very small. But if you can amplify it properly, you're going to be able to carry out the detection very early. So for detecting viral infection, short sequences that are complementary to the viral genome are used as the primers, right? And following many cycles of amplification, the presence or absence of even a single copy of a viral genome in a sample of blood can be ascertained, right? And once the amplification has been carried out using PCR, the virus detection can be done using gel electrophoresis, right? Subject it to an electric field and see where it goes, right? Based on that, you can see whether the person has the virus or not. And the schematic diagram for the entire procedure in the case of the HIV virus, which causes AIDS, is shown in the next figure, right? So you can see you're going to take blood sample from the infected person, right? Then you need to extract the viral RNA, or in the case of AIDS, it's going to be RNA. So you're going to remove the cells by centrifugation. Remember, I covered this when I talked about proteins. Okay, at high speeds, you centrifuge, you separate out the cellular components, right? Then you'll get a rare HIV particle if the person is infected in the serum of the infected person, right? So now you're going to extract the viral RNA genome, right? So get rid of the protein coat of the virus, you got your RNA. Now use reverse transcription to get the complementary DNA, right? Now make it double-stranded, again by using DNA polymerase. So now you got like one copy of that DNA that you're interested in. Before you can run any procedures like gel electrophoresis, you have to amplify the amount of DNA, right? So for that you can use PCR. So PCR amplification will give you lots of copies of this. Now you can run these lab procedures, all right? So you have this gel electrophoresis here. So in one lane you're going to run with the blood from the potentially infected person, right? And see where it moves. In the other lane, right, you're going to run, you're going to take blood from the normal person, subject it to exactly the same procedures, see where it moves, right? And then looking at, comparing the two, you'll be able to figure out whether that person is infected or not. Okay. All right, any questions? So now I'll talk about applications of PCR in forensic medicine, right? So PCR has got great potential in forensic science used to track down the perpetrator of a crime. Now the DNA sequences which create the variability used in this type of analysis, they contain runs of short repeated sequences such as GT, 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 which are found in various positions or called loci in the human genome. All right. So in our genes, we have these repeated sequences. All right, in some positions. All right, and everybody has a different number of repeats. All right. So the number of repeats in each run is highly variable in the population, ranging from as few as 4 to as much as 40. Right, in different individuals. So a run of repeated nucleotides of this type is commonly referred to as a variable number of tandem repeat sequence. Variable because it varies among members of the population. Right. So because of this variability in these sequences, each individual will usually inherit a different variant of each VNTR locus from their mother and from their father. And therefore, two unrelated individuals will therefore not usually contain the same pair of sequences. Okay, but at least if you're related, probably there will be some similarity. Some of the sequences will be similar. So that's how you can track down relatives also. Right. So when you're doing forensic science, all right, a PCR reaction using the primers that bracket the VNTR locus produces a pair of bands of amplified DNA from each individual, one band representing the maternal variant and the other representing the paternal variant. Right? And the length of the amplified DNA will depend on the exact number of repeats of the locus. Right? So the position of the, and again, as I said, before, when you're when you're running gel electrophoresis, all right, on a DNA strand, on a DNA fragment, the length of the fragment decides the location to which it will migrate, right? So if you have different number of repeats of those GT, all right, the fragment will be of different lengths, all right? So they will migrate to different locations, all right? So the position of the amplified DNA after gel electrophoresis will depend on the number of repeats that are present at a particular locus. This fact can be used to narrow down the list of suspects in a criminal investigation. Right? And let's look at the detailed steps. So the first step is you're not going to use just one VNTR locus, okay? Because that's like looking for a needle in a haystack. Okay? So you'll take a number of these VNTR loci, right? You'll select a number of them. You will use the primers to bracket each locus because you don't want to amplify all of the DNA. Only in that locus, right? Or in those loci, you want the DNA to be amplified. So the DNA is going to be amplified using PCR, and then you run gel electrophoresis. So this process should be carried out on DNA that is obtained from the forensic sample and also DNA from each of the suspected individuals. Right? So if the bands obtained during gel electrophoresis on the forensic sample do not match the bands obtained from a suspect, all right? If there is no match, right, then you know that this particular suspect can be reasonably eliminated from the list of suspects. Right? But if there is a match, does that mean that that person is the suspect? Not necessarily, right? Not necessarily, but one can do calculations to calculate the probability that this is the person. Okay? And if you find that the probability is something like 1 over the population of this whole world, right? Then I mean, with a reasonable degree of confidence, you can conclude that this is the guy that committed the crime, right? Although his or her lawyer will probably try to find holes in that argument too, right? And that's what these highly paid lawyers, right? They'll try to tear that thing apart, right? Because it's not 100% certain. Okay, with very very high probability you can say that this is the guy. But at least for exonerating people, it's going to work, right? For ruling people out, it's going to work because if the bands don't match, then it cannot be that person. If they match, it could be somebody else's bands also might match, but it's very very unlikely. All right, any questions? So next we move on to the chapter on genetic engineering, right? And here, like there was an earlier chapter where we talked about genetic variation, right? That's stuff that happens in nature, and it happens randomly, not in a predictable manner. Okay, it's just by chance, right? In genetic engineering, we are going to engineer the mutations, right, and produce different kinds of organisms. So advances made in DNA technology over the last 3 decades, actually it's 4 decades now, right? Because this has been since the 70s, right? So have opened up vast avenues for genetic manipulation. For example, using DNA technology, completely novel DNA molecules can be constructed. To do so, one can cut a plasmid vector using a restriction nuclease. Again, we talked about this before, and then insert a DNA fragment into the plasmid vector using DNA ligase, right? Stitch it back, cut it, then stitch it back. the procedure can be repeated to insert additional DNA fragments. Another application of DNA technology is the production of large quantities of rare cellular proteins using cloned DNA, right? So to do so, one can make use of a plasmid that already has a promoter sequence inserted into it, right? And such a plasmid is called a double-stranded expression vector, right? And then after that promoter region, you can put in the DNA sequence of interest, right? And then use it to produce protein. So the protein coding sequence is inserted into the region immediately following the promoter by using a restriction nuclease and DNA ligase. Again, the nuclease is for cutting, all right? Ligase is for stitching back, right? Then by introducing the recombinant plasmid into bacterial cells, these cells are going to be overexpressing the messenger RNA, right? And the corresponding protein, right? So even if a protein were rare, now the bacteria is going to be producing a lot of that protein, right? Yet another application of DNA technology is the artificial production of RNAs by in vitro transcription. You can carry out transcription inside the test tube, okay? Because see, unlike DNA, RNA is not a stable molecule, right? RNA is a temporary copy that is produced on an as-needed basis, right? And it is easily disintegrated. You have to go to great lengths to preserve RNA. For example, on our skin, right, on the nose and all that, we have RNA disintegrating enzymes, okay? Why? Because if a virus invades or bacterium, right, we shouldn't fall sick that easily. So the body has got defenses, okay? So they have RNAs. So if you just go and touch RNA, right, it's all messed up now, right? You cannot use that for experiments anymore. You might have to freeze it at like temperatures like minus 80 degrees Celsius and things like that, okay? DNA, you don't have to do any of that, right? So most RNAs that are found in a cell are present only in small quantities, and they are hard to isolate because it's very, very sort of fragile molecule, right? So it's hard to isolate from the other cellular components. So once the gene coding for an RNA has been isolated, that RNA can be produced artificially in a test tube by letting RNA polymerase operate on that gene, right? Not inside the cell, but you can produce large quantities of that. Now the function of a gene is best revealed by an organism that has had that gene mutated in some way, right? Because see, if you're looking at the human genome, right, you have like 3 billion nucleotides. You have like 20,000 plus genes, right? I mean, how do you figure out the function of a particular gene, right? Maybe that gene just doesn't act by itself. Maybe it produces some protein. Some other genes produce some other proteins. Like you have those transcription factors, and it's the combination together, okay, that functions as a unit. So how do you figure out the function of this gene? Just looking at the sequence is not enough, right? So neither the complete nucleotide sequence of a gene nor the three-dimensional structure of a protein is sufficient to deduce a protein's function, because the protein may not be acting by itself. It might need to bind a lot of other factors, right? So many proteins, such as those that have a structural role in the cell or normally form part of a large multi-enzyme complex, will have no obvious activity when viewed in isolation. So on the other hand, if you have a mutant organism that lacks that protein, right, that might clearly reveal what the normal functions of that protein are, right? For example, if you have a mouse that lacks the gene for insulin, okay, that mouse will be a diabetic mouse, right? Right? Because you will see that the organism is not able to metabolize sugar properly, right? Now, prior to the advent of recombinant DNA technology, such mutations would have had to arise naturally, and so would necessarily arise by chance and be unpredictable. Like today in the lab, you can produce a diabetic mouse, right, just by taking that gene for insulin away. But in the past, what would happen is you'd have to wait for the mouse to grow, old and naturally get diabetes, right? So recombinant DNA technology makes it possible to introduce precise mutations by the technique of site-directed mutagenesis. And let's see examples of that, right? So here what you've done is that you have the plasmid cloning vector, right? Plasmid again is a small double-stranded circular DNA molecule, right? So here you've inserted the normal gene, right? Again, by using, what did you use to insert the normal gene over here in the plasmid? Right. So you use restriction nuclease to cut it, then you put that thing in, and then you ligate it, you stitch it back using DNA ligase. So this is the normal gene, all right, that you have put in this plasmid. Now what you could do, all right, is when the two strands separate out, you could separate out the two strands, and instead of putting the normal gene, you could put in a mutated gene in the bottom one, right? See, this was GAC, that is complementary to CTG. You basically change the base in the middle, right? You've changed it to GCC. Okay, so it's not a perfect hybridization because there's something sticking out here, right? But now look what has happened, right? And then you let the two strands get back together. So it's still sticking out. But now if you're going to replicate this plasmid DNA, right? Every time it replicates, right, each strand is going to be used as a template, right? So if the outer strand is used as a template, you're getting the correct, DNA sequence on the inner strand. If the inner strand is being used as a template, then this stuff is all messed up, okay? But messed up in a very, very precise way, right? So now every time this thing divides, all right, the cell divides, the plasmid DNA is getting replicated. Then on the left, okay, you have the correct sequence. So you have GAC when the messenger RNA is being transcribed, let's say from CTG, you're getting the correct one. But on this one, you're getting something else, right? And this GAC, it codes for the amino acid asparagine, right? Whereas GCC codes for alanine, right? We have not done these codes here, but if you look in one of the books, you will see that, okay? So it's a different amino acid, so the property will be different. So now you have created site-directed, very precise site-directed mutagenesis, right? Where here you have, one sequence, and so half of the progeny will have one sequence, the other half is going to have the other sequence, the mutated sequence, right? And it has been carried out with a lot of precision. Okay, nothing has been left to chance. So again, I mean, this is the same stuff written out in the text. So in recombinant DNA technology, one can start with a cloned gene and proceed to make mutations in it in vitro. Then by reintroducing the altered gene back into the organism from which it originally came, one can produce a mutant organism in which the gene's function may be revealed. For example, in that picture, the gene to be mutated is first inserted into a plasmid, and a sequence GAC, say in one of the DNA strands in the gene, is replaced by a mutated sequence GCC. That was in the inner strand, all right? And then the plasmid is introduced into a cell. Due to the semi-conservative nature of DNA replication, every time the cell replicates, one half of the progeny end up inheriting the normal gene, while the other half end up inheriting the mutated gene, right? And the result that half of the progeny will produce the corresponding normal protein, while the other half will produce the mutated protein, right? So this is stuff that you can see, all right, what's going on in the organism, right? Now, organisms that have the DNA in them altered in some way, they are called transgenic organisms, right? So recombinant DNA technology can be used to carry out gene replacement. You could replace one gene by another, right? You could produce gene knockout. Knockout means you make it dysfunctional. You knock the gene out. Or you could put in an extra gene, right? If you want to, produce for that animal to produce more hemoglobin or insulin, you can put in an extra copy of that gene. So organisms into which a new gene has been introduced, or those whose genomes have been altered in other ways using recombinant DNA techniques, they are known as transgenic organisms. Right? So when the biologists they do research, all right, they have to get special permission. If you are using recombinant DNA, all right, transgenic animals and all that stuff, if you submit a proposal to a funding agency, before you can get the money, they have to make sure that all your protocols are proper, right? Like you cannot say that I'm a sadistic person and just for the fun of it, I'm going to create a 10-headed dog or something like that, using DNA technology. They'll not let you do that, right? And even if you have to, let's say sacrifice animals, right? For the purpose of research, you have to do things in a humane way to show that you're going to make it as painless as possible, because they inject carcinogens into animals to make them get cancers, and then they want to see the treatment and all that, So you have to have a proper experimental design and all that stuff. Now, when you're trying to engineer transgenic animals, it is clear that for haploid organisms, that means organisms which have only one copy of each gene, such as bacteria or yeast, transgenic progeny can be produced quite easily, right? Just like we already discussed, all right? Right? I mean, the putting it in the plasmid and getting, because your job is basically to mutate only one copy of that gene, because half of that progeny, when you put in those plasmids, half of that progeny are mutated, okay? The other half is not mutated. So here it's easier because you have to do only on one chromosome, okay, or only one copy. But if you're trying to create a diploid organism, right, then you have to make sure that the mutation happens on both the copies, okay, that is more difficult. So for diploid organisms such as mice, it is more difficult but still possible to achieve gene replacements. This involves the following 4 steps. See, if you're going to do if you're going to produce a mouse with gene replacement, okay, you cannot go and pick an adult mouse and try to change every cell in that mouse, okay. That's a lost battle. You cannot do it. You can do it for a few cells maybe, all right. So the way you have to bring about gene replacement in a mouse is basically you will have to intervene at the stage when that mouse is like an egg, all right, fertilized egg or sperm. You have to put mutations in those, okay. So it's a little bit more complicated. So here this involves the following 4 steps. An altered version of the gene is introduced into cultured embryonic stem cells, right. Now few rare embryonic stem cells will have their corresponding normal genes replaced by the altered gene through homologous recombination. We all know what homologous recombination is, okay. Sequences which are similar, they line up together and then there's a crossover, right. Although often laborious, these rare cells can be identified and they can be cultured to produce many descendants, each of which carries an altered gene in place of its 2 normal corresponding genes, okay. So these altered embryonic stem cells, some of them, I mean, at least the altered ones, they have that gene mutated. They are injected into a very early mouse embryo. As a result, the cells are incorporated into the growing embryo, and a mouse produced by such an embryo will contain some somatic cells, some cells in the body, right, that carry the altered gene, right. Now some of these mice, again, I mean, there is stuff that is left to chance, okay, because only some of the cells will get that, okay, and some of these mice will also contain germline cells, right, that is the precursors of the reproductive cells, right, the cells from which the egg and the sperm are produced, right. Some mice might have mutation in that. That is how it is going to get transmitted to the next generation, right. So now you'll identify mice that have these germline cells with that mutation, right. So when a mouse of this type is bred with a normal mouse, some of the progeny of these mice will contain the altered gene in all of their cells, okay. Because you know, if there is a mouse that contains germline cells which contain the altered gene, okay, so now out of 2 copies, one of them is altered. So the egg or the sperm that is produced from that organism, right, some of them will have that mutated gene, right. So now if that egg or sperm with the mutated gene fuses, right, with let's say another normal sperm or egg, right, the organism that will be born from that, right, is going to still have one mutated copy of that gene in each and every cell of the body, right. So when bred with a normal mouse, some of the progeny of these mice will contain the altered gene in all of their cells. Again, some of the progeny, why? Because you know, if that person has got, or if that mouse has got one mutated copy, right, it is possible that they will produce also perfectly normal egg or sperm, right. So in that case it will not work. So when bred with a normal mouse, some of the progeny of these mice will contain the altered gene in all of their cells. This is because 2 haploid reproductive cells of different kinds, egg and sperm, have to fuse together to produce the diploid cell that will undergo repeated cell division to produce the entire organism. But now you want a mouse that has both copies knocked out, right. So there's further breeding that has to take place, right. So if 2 such mice are in turn bred, okay, so there is a mouse that has every cell in the body having a mutated gene, right, 1 copy. Another mouse that has every cell in the body with a mutated gene, right. Then if you breed the 2 of them, then there is a 1 in 4 chance that the progeny will have, you know, both copies knocked out, right. So if 2 such mice are in turn bred, some of the progeny will contain 2 altered genes, 1 on each chromosome in all of their cells. This again is a consequence of the meiotic process by which the haploid gametes are produced from their diploid precursors, and the fact that when the egg with an altered gene fuses with the sperm with the same gene altered, then the resulting diploid cell will have both copies of the gene altered, right. And if the original gene alteration completely inactivates the function of the gene, then we will have what are called knockout mice, right. You're doing a knockout, right, gene knockout. You knocked out the gene for let's say insulin or something like that, right. And these organisms are actually commercially available, okay. You can place an order, you can tell them, okay, send me 50 mice with this gene knocked out, okay. They'll charge you quite a bit of money, but they'll send it to you, right. If you don't want to go through the procedure and all this breeding and all that stuff, yeah. So I think we are done with this chapter for today. Let's stop here for today.